Welcome to the participant story. Real people, real voices, and real experiences from inside clinical trials. In each episode, we sit down with a different guest to hear why they volunteered, what they experienced, and how participation in clinical research has changed their lives. The participant story is brought to you by Curevit Clinical Research. Curevit leverages technology, local healthcare infrastructure, and decentralized clinical trial elements to design and execute clinical trials where participants come first. Learn more about Curevit and their innovative virtual clinical site at curevit.io. The participant story is hosted by industry veteran and entrepreneur Dave Hanneman.
Hello, everyone. I'm excited to introduce today's guest, Dr. John Woog. He's a remarkable physician, surgeon, and patient advocate whose story bridges science, medicine, and hope. Dr. Woog began his journey at Penn State, where he received the President's and Evan Pugh Awards before graduating valedictorian from Jefferson Medical College. He went on to train at some of the world's top institutions, completing an internship at Johns Hopkins, an ophthalmology residency at Harvard's Massachusetts Eye and Ear Infirmary, and a fellowship in oculofacial surgery at the University of Wisconsin-Madison. Over his career, he's held leadership and teaching roles at Harvard, Tufts, the University of Wisconsin, and the Mayo Clinic, where he served as professor and consultant in ophthalmology. He's earned multiple Teacher of the Year awards and recognition for outstanding patient care. Dr. Woog is also a prolific researcher and author with more than 65 scientific papers, 30 book chapters, and 3 books to his name. He served on the editorial board of Ophthalmic Plastic and Reconstructive Surgery and received leadership and achievement awards from the American Academy of Ophthalmology and the American Society of Ophthalmic Plastic and Reconstructive Surgery. But what makes Dr. Woog's journey so powerful is what came next. After being diagnosed with stage 4 cancer, he faced years of surgeries and chemotherapy, and ultimately had a dramatic recovery through an experimental therapy. That experience transformed his mission, inspiring him to dedicate his life to patient advocacy and to helping others navigate serious illness in clinical trials. Together with his wife, he co-authored Facing Cancer with Hope, sharing lessons and encouragement for patients, caregivers, and advocates. He continues to speak nationally at the Mayo Clinic Center for Individualized Medicine, ASCO, and the Hope for Stomach Cancer Summits, and serves as a certified patient advocate and subject matter expert for the Patient Advocate Certification Board. In 2025, Dr. Woog joined the ASCO Tapur Study Steering Committee, working to expand access to innovative cancer therapies and advance patient care across the country. Please join me in welcoming Dr. John Woog, a physician, survivor, and advocate who continues to inspire through his expertise, compassion, and unshakable hope. Good morning, John.
Good morning, Dave.
It's really a pleasure to have you today. As you know, the Participant Story podcast, our intention is to find people who have participated in clinical trials and hear their story. And that's really it. Our desire is to get people's stories out in their own words, let them take this story where they wish to. And my role, as I see it, is simply to facilitate that journey. I feel particularly privileged to have you on today, as your bio mentions. Not only do we have a participant, but we also have a physician and a researcher all in the same package. So thank you so much for taking time out of your day today and sharing your story with me, but also with all of our listeners.
Oh, it's my privilege and a pleasure, Dave. Thank you.
So before we jump into it, John, where do I find you this morning?
So we're actually in the upper Midwest in Rochester, Minnesota, where We're now at September 3rd, and we did have a little bit of frost last night. So just for folks watching from other parts of the country, give you a little taste of early Midwestern fall here.
Great part of the world and a great time to be there. So thanks again for joining.
Oh, our
Uh,
pleasure.
Let's jump right into it. John, I understand you and your family have faced a major medical challenge. Can you tell us a little bit about it, about your background and your initial diagnosis?
Sure, Dave. So I'm an ophthalmologist by training, and I was in my mid 40s. This was some years ago now. We had two young kids. I was an active surgeon, teacher, administrator in our eye department. I was also a marathon runner, not a very fast marathon runner, but I'd say a plodding runner. But I noticed it was getting a little more difficult to train. I was a little more fatigued. Um, I frankly just attributed it, to age, which catches up to all of us, certainly. I was having some abdominal pain, and for that reason, ultimately ended up having a procedure called an EGD, or an upper GI endoscopy, which really, to our collective shock, my providers and my family and myself, showed that I had advanced stomach cancer.
Mm. Well, that must have been quite a shock. Tell me a little bit more about stomach cancer. As I understand it, it's not particularly common, at least in the West.
That's absolutely correct, Dave. Stomach cancer is almost considered a relatively rare cancer in the West. In the U.S., there are about 30,000 new cases diagnosed each year. This represents numerically only about 1.5% of cases of new cancer diagnosed in the U.S. This contrasts with approximately 320,000 new cases of breast cancer that are diagnosed each year here. So really a tenfold difference in incidence. Stomach cancer doesn't even rank, in fact, in the top 10 diagnoses of new cancer in the U.S. Each year. Now, the picture is much different worldwide. There are close to 1 million new cases of gastric cancer diagnosed each year. Stomach cancer is actually worldwide, again, the 5th most common cause of cancer. The incidence of stomach cancer is particularly high in East Asia, countries such as Korea, Japan, and China. It's, in general, I'd say it's a difficult form of cancer to treat, particularly when the diagnosis is made at an advanced state, which again is unfortunately very common in the West. It represents, in addition to being the 5th most common type of cancer in the world, is actually the 5th, some folks say the 4th most common cause of cancer mortality or death worldwide.
That's sobering. How is it usually diagnosed here or in Asia?
Well, a great question, David. So one of the challenges, again, here in the West, in the U.S., we don't have at this point a regularly administered screening test for stomach cancer, unlike breast cancer, prostate cancer, colorectal cancer, now even lung cancer. We really don't have a readily available screening test. Again, for that reason, it's usually advanced at the time of diagnosis with and diagnosed really based on symptoms such as mine, abdominal pain, weight loss, difficulty eating, feeling full earlier while eating is another symptom that unfortunately is more often recognized in retrospect. In East Asian countries, it's a kind of a different situation due to the higher incidence of gastric cancer in those countries. They have adopted regular screening programs, often incorporating, again, this upper endoscopy or EGD procedure. This is analogous, I would say, Dave, to colonoscopy, which is, again, recommended here, or other now non-invasive forms of colorectal cancer screening, which are recommended by public health authorities in view of the high incidence of colorectal cancer here. So it's kind of almost a flip situation
Hmm.
there.
Interesting. So returning to your experience, what happened after your diagnosis?
Yeah, so by the time I was diagnosed, I had actually lost about 10 pounds. I thought maybe I was from training, but in retrospect, really, I wasn't eating as much. I was having difficulty finishing a full meal. I was actually anemic on my blood testing and had some other blood indices of malnutrition. Due to my difficulty in eating, the decision was made to actually proceed directly with surgery, which I had a week after my diagnosis. I should note that now, and again, it's a good number of years later, things have changed in terms of the protocols for management of gastric cancer. And in many cases, when possible, chemotherapy is actually administered prior to surgery in an attempt to shrink the tumor before the procedure. But for me, it was straight to surgery. The surgery itself, sorry if I'm being a little bit graphic, and my apologies to any listeners or members of the audience who are about to sit down to a nice breakfast or lunch, but my surgery consisted of removal of my entire stomach, the bottom part of my esophagus, and then part of my small intestine to which the stomach connects. My spleen was involved due to the large size of the tumor. It actually invaded my spleen, so that had to come out as well. Typically, during surgeries of this type, the surgical team also removes many, many, many lymph nodes to look for the evidence of cancer spread. My team removed, if I recall correctly, about 50 lymph nodes draining this area, following which they then ultimately performed a reconstruction, which involves moving parts of the remaining small intestine around and suturing it to the esophagus. So after the surgery, the removed tissues were examined under the microscope by the pathology team. The good news was it looked like the surgical team had gotten around the entire tumor. There was no tumor at the edges of the very large resected specimen. Unfortunately, well over half of the resected lymph nodes did show evidence of cancer. And this was not good news, Dave, as data at that time suggested that only about 27% of individuals with the degree of lymph node involvement I had survived for 7 years or longer.
That sounds like both a brutal surgery and tough news. What did you do next?
Well, yeah, it was definitely a major surgery, Dave. I was in the hospital for about 2 weeks afterwards. Big shout out to nursing team, all the providers who help the folks get through these bigger surgeries. We then did have a discussion with my medical oncologist due to the extensive lymph node involvement. It was recommended that I proceed with postoperative chemotherapy to try to kill any remaining cancer cells. So I had the 2 weeks in the hospital. I had 6 weeks after the hospitalization for recovery purposes before starting chemo, and we used that time to investigate the different options in terms of chemo.
I imagine as a physician that you've got quite an extensive network of colleagues, friends, peers. You mentioned earlier that stomach cancer is particularly common in Asia. Did you reach out, receive any guidance from clinicians or scientists in Asia?
You raised a very perceptive point, Dave. As a physician, I've been extremely fortunate to have really great sources of advice. And in fact, yes, we emailed stomach cancer specialists throughout the U.S., Europe, and then certainly several very experienced gastric cancer oncologists in Korea and Japan. As you mentioned, I'm very fortunate. This process was facilitated by pre-existing professional friendships, which I had with eye surgeons in Korea and Japan, and they provided us with the names of some folks to contact. I have to say we were absolutely humbled by how so many specialists here in Europe and Asia were so generous with their time and their advice.
Well, so what did they recommend, and did it differ at all from what you had learned in the U.S.?
Yep. So the consensus recommendation, first and foremost, was that indeed, yes, I needed chemo. The normal type of chemo administered, and I'm sorry to get a little bit into the weeds here, for many types of GI cancer, including colorectal cancer, was a combination of drugs called 5-FU and oxaliplatin. Both of these drugs are in the category of what are called cytotoxic chemotherapy agents. That means that they kill cells, and they're also non-targeted, meaning they kill both normal cells, particularly the rapidly dividing cells that we all have in our bodies, in the GI tract, our blood system, and skin, as well as the cancer cells. So these drugs are generally both given IV by intravenous infusion. They do, as we mentioned, they do impact normal tissues as well, and that explains the significant incidence of associated side effects, adverse effects, ranging from nausea and vomiting to anemia, bleeding, and even infection if the white blood cell count is suppressed. At the time of my diagnosis, there was actually a lot of excitement in Asia about a new oral drug as opposed to an IV drug called S1. And S1 is what's called a prodrug, meaning it's a drug that we take and then it's converted by our enzymatic systems within our bodies to an active chemo agent, in this case, actually 5-FU. So the drug, in addition to being oral, which was nice, was formulated to reduce the incidence of some of these off-target adverse effects that we mentioned. Again, a big advantage from the patient perspective was that since the S1 was a pill, I wouldn't need the IV infusions of 5-FU, which actually take 46 hours each. It's quite a long process. So based on this collective feedback, it was decided that I would use a combination of this new drug S1 and oxaliplatin.
How long were you on chemo, and how did you do on it? What came next?
Yep. So I was on the S1 oxaliplatin regimen for about 4 months. I'd say in general, I tolerated it pretty well. I, again, have to give a huge shout out to the oncology nursing staff in the ambulatory center where chemo infusions are given. They're absolutely amazing. They provide each patient receiving chemo a detailed orientation to the process, what to expect, what the danger signs are, et cetera, associated with chemo that should prompt a call or a visit to the emergency room. The nursing staff and my oncologist as well, who I saw about every month, I was in weekly for the chemo infusions, but I was monitored very closely during our visits to ensure that any potential issues were hopefully picked up and remediated quickly. So anyways, that was 4 months of chemo. I finished the regimen in the early summer. I was diagnosed, I should have mentioned, back in January. So surgery, healing, chemo, it's already, getting into the late summer. Our family had not availed ourselves of any vacation during what Dave, is a really short upper Midwestern summer season. And in an attempt to do that, we took a little road trip. That went great. We had a great time. I returned from my vacation refreshed and chomping at the bit to get back to work after Labor Day, which was coming up. I was still having some discomfort while eating, and we decided to get an abdominal CT scan to look at my GI tract. As it turned out, my GI tract was absolutely fine. I was shocked, however, to learn that the CT scan showed a punched out lesion in the liver consistent with an area of metastasis from my stomach cancer.
Oh my, that must have been surprising and rather brutal news.
I would say,
What happened
Dave,
next?
was probably harder news in a sense to get than the initial diagnosis, which was not good news or pleasant news. But having gone through the surgery and the chemo and really literally looking forward to getting back to work the next week, it was very discouraging. We knew from the scientific literature at that time that the median survival for folks with metastatic gastric cancer with liver involvement was only 7 to 15 months, despite any available additional treatment. So again, yes, very discouraging news at that point.
Well, discouraging and so surprising based on what seems like it was pretty good news in terms of the original concern. So what did you do next?
So we spent the Labor Day weekend looking up different treatment options. Thank goodness for the internet, Google, PubMed at the time, and clinicaltrials.gov, ultimately, as we'll discuss. Early the following week, we met with my oncologist, and I still remember that conversation now 17 plus years later, as was his responsibility based on current data at that time. He very gently indicated to my wife and me that the goal of further treatment at that point was now, in a sense, buying some more time, as opposed to the hope for cure that we had with the initial surgery and chemotherapy. Based on the disappointing results for individuals with metastatic stomach cancer involving the liver, again at that point, 7 to 15 month median survival, we decided to pursue what was then a relatively new approach towards a treatment, namely targeted therapy. In targeted therapy, treatments are directed against specific characteristics of the cancer that are relatively distinct to the cancer, or specific biologic pathways that the cancer is using to survive, to grow, and to spread. I think at the time of my diagnosis, again now about 18 years ago, there were only 3 targeted agents available, approved by the FDA for cancer treatment. The first drug, and again, I'm sorry for getting in the weeds a little bit. I know you have a very sophisticated audience, Dave. But the first drug was one called rituximab. This was approved in 1997 for treatment of certain types of lymphoma. That was followed the next year in 98 by trastuzumab, which was approved for treatment of a certain type of breast cancer called HER2 positive breast cancer. And then finally, in 2003, just a few years before my diagnosis, a third targeted agent called imatinib was approved to treat a form of small intestinal cancer. At that time, there were no FDA-approved targeted therapies for stomach cancer, and that meant my main access to targeted agents would be through a clinical trial. Given the disappointing outcomes that we would anticipate with conventional chemo, my oncologist agreed with pursuing a clinical trial. Although I would say as an ophthalmologist, I had helped care for individuals who had been on trials in the past. This was really my first up close and personal exposure to the trial process.
Interesting. With your background, experience, education, you're sort of the perfect example of an informed patient. And yet this is now your first personal experience in a trial. Was the decision to participate straightforward for you or difficult in any way?
Yeah, so good question, Dave. Actually, with that data regarding the poor historical outcomes, again, the median 7 to 15 month survival with additional chemo, that actually made the decision to pursue a trial of a novel agent, a new therapy, pretty straightforward, I would say. Deciding which trial to pursue, though, was a little bit more involved.
So how did you select the clinical trial that you ultimately participated in, and what was the decision-making process to select between trials?
So, as we mentioned, we're very grateful for the availability of the clinicaltrials.gov website. We went to the site, typed in gastric cancer in the search bar. A number of studies came up. Some of these studies, and I think this is true for many types of cancer, are observational, and they're directed towards collecting data about individuals with a particular type of cancer. And these studies are certainly worthwhile, they're certainly important, but the goal of those studies was not aligned with my particular need for a new direction in terms of treatment. Similarly, there were other very worthwhile studies listed that basically looked at varying combinations, incremental changes in the delivery of conventional chemo. And overall, I didn't think that those types of studies were likely to be as impactful as we hoped either. So we therefore decided to narrow down the search and to trials that were specifically mentioned targeted agents. Our process of selecting the particular trial was a lot different then than it is now. Currently, the process of selecting targeted therapy for cancer begins with a review of a number of assays that look at the genetic, the protein composition, other molecular characteristics of the tumor. And these assays kind of point the way towards the pathway or the molecule that one wants to target. Back when I was diagnosed, Dave, again, over 18 years ago, really most of these tools were not available. We did have one tool available, again, sorry to get a little technical, called karyotyping, that evaluates the number and integrity of chromosomes in our cells. Now, chromosomes, for a brief Bio 101 review here, they're the structures in our cells that house the majority of the 23,000 or so genes that we all have in our cells. The genes, in turn, code for the proteins that are the metabolic workhorses of the cell. They allow basically the fulfillment of the functions of life, and unfortunately for cancer cells, these proteins propel, again, growth, spread, and division of the cells. So getting back to the chromosomes, each of our normal cells has 23 pairs of chromosomes. Karyotyping of my cancer tissue revealed that there were extra copies of one particular chromosome, chromosome 7. Now, this was interesting information because at that time there were just some publications coming out linking abnormalities in genes on chromosome 7 with a number of types of cancer. One gene called MET, M-E-T, had recently been associated, again, as it turns out, with a number of forms of cancer, including gastric cancer. So with that information in mind, we ended up narrowing our search on clinicaltrials.gov to trials that targeted this MET pathway. And we did indeed, we're happy to find a study investigating a new therapy targeting MET. This drug was what's called a small molecule, similar to the drug mentioned earlier, imatinib, that had been proven actually very effective in the management of small intestinal cancer. So we were hopeful in general that this small molecule might have a similarly beneficial effect in terms of inhibiting MET. In reading more about the trial, and again, I'm learning about not only the biochemistry of cancer at this point, but about the clinical trials process, Dave. We learned that it was a phase 2 trial. As I'm sure your very informed audience is aware, phase 1 trials in oncology generally focus on demonstrating safety and also on determining what's called a maximal tolerated dose for the agent. In some cases, and fortunately this is happening more often now with some of the recent trials of immune active agents, you get some indication during phase 1 trials of efficacy, effectiveness as well. At that time, back again 18 years ago, that was not a tremendous expectation. But it was good to learn from the patient's perspective that the phase 1 study of this drug revealed no major safety concerns. Sometimes safety issues don't show up in phase 1 studies, even in the larger phase 2 and phase 3 studies. Sometimes they don't show up until after the drug is approved and on the market for a while. But nonetheless, the information from the phase 1 study was at least encouraging from the safety perspective. Also, again, from the patient's perspective, since this was a phase 2 study, it was an open label study, meaning I knew that I was going to get the study drug, as opposed to many phase 3 studies where individuals are divided into 2 arms, one of which gets so-called standard of care treatment, and the other of which gets the experimental treatment. And at this point, certainly my goal in participating was to get an active agent. So the fact that this was guaranteed in the phase 2 study was also a positive.
So it seems like you sorted through your options in a very methodical way. Were there other factors to consider in evaluating the trial? And did the trial raise any particular concerns or questions before you decided to join?
So in terms of other factors, Dave, yes, I was looking for a trial in a major medical center that had experience in caring for individuals with gastric cancer. So that was one consideration, certainly. As it turns out, my primary oncologist and personally knew the principal investigator, the oncologist running this trial, and knew that this individual also had a lot of experience, or as much as experience in the U.S. as really anyone has with gastric cancer. In terms of my concerns, I'd say my wife and I did have some questions regarding the study logistics. I'd have to say, though, our overall feeling at the point was we were very grateful that there was an option to explore beyond more conventional chemo, which we knew really wasn't very promising. So overall, we approached participation in the trial very positively.
So you've at this point been a physician, a researcher, a teacher. Now you're a participant in a clinical trial. Tell us a little bit about what participation felt like for you.
Yep. So first, Dave, in terms of logistics, the nearest center for the trial was in Chicago, which is about 350 miles or so from our home in southeast Minnesota. After my oncologist and my wife and I decided that the trial was a reasonable option, the next step was my oncologist sent a lot of medical information, test results, pathology results, et cetera, to the clinical research coordinator in Chicago for review. Review of all this information confirmed that I met both the inclusion and exclusion criteria for trial participation. So my wife and I flew to Chicago. I think we stayed, if I remember correctly, overnight in a hotel prior to visiting the medical center there. The next morning, if I remember correctly, our young kids at the time were very grateful to have the support of our family members who kept an eye on the kids. At the hospital, we met with the clinical research coordinator and the principal investigator and went through a appropriately very lengthy, detailed, careful, informed consent process. I then had extensive baseline testing at the medical center there. Included blood work, an EKG, an echocardiogram to look at my heart function. I was then given the bottle with the study medication. The instructions were to take 1 pill every day for the first 5 days of each 2-week treatment cycle. At the end of that 2-week cycle, I would return to Chicago for follow-up testing and provision of another supply of medication for the next cycle. I have to say the experience throughout from the patient perspective, all the staff we interacted with, Dave, from folks giving us directions from the reception desk to the allied health staff, the nursing team, again, fantastic, clinical research coordinator and the physician team in the cancer center in Chicago, they were all wonderful, compassionate, very professional, and again, I think provided significant support to my wife and to me as we were embarking, as you mentioned, on what was really an uncharted journey for us.
Praise and appreciation for staff is a consistent theme on these podcasts. It sounds like there was a fair amount of travel involved. Am I correct? If I'm
Yes.
thinking about my geography.
Yep. Absolutely, Dave. There was after our initial visit, my wife and myself. I made most of the study visits by myself. On a typical visit day, I'd get up about 4 a.m, drive to our local airport to catch a 6 a.m. Flight to Chicago. It's about 1 hour flight. It actually took longer than the flight duration, about 2 hours or so, to get from Chicago, the airport there, to the medical center. And this involved a combination of the subway, the L, and cabs. And I have certainly, based on that experience, I had newfound respect for folks who lived in Chicago and navigated big city challenges there. At any rate, at the medical center, I would get blood studies drawn, other required tests, including, in some cases, repeat EKG or echo. Sometimes I'd actually have several blood draws during the course of the day to study the metabolism of the study drug. Again, these are new drugs not approved for use, and it's important. Study sponsors and the FDA really require information about how the body metabolizes the drugs. So that was part of the study as well. Overall, my personal goal was to be able to get back to the airport after all the testing in time for what was actually the last flight out, so to speak, from Chicago to Minnesota, so I could see my wife and kids before they went to sleep. Most of the time, I would say that traveling went okay. It was not over the winter months, fortunately, but my wife did return from one trip, and we did have one trip where the airport in Chicago was actually closed due to a sudden weather event unforeseen while we were in the air, and the plane was diverted to an airport 3 hours south of Chicago, and it ended up taking a combination. It's like that movie, if you've ever seen Planes, Trains, and Automobiles.
Yes.
It took a combination of buses, trains, and cabs to ultimately get to the medical center in Chicago in what ended up being a 9-hour journey. So, again, kudos to the team, the study team, the hospital team there. They very graciously were beyond their hours and accommodated us, did the testing, and provided us with a new medication. I would have to say overall, Dave, I was extremely fortunate on many levels to have the ability, frankly, the financial resources, since travel was not covered by the study sponsor, and the stamina, what's called in oncology terms, the performance status, to make the trips to Chicago. But this experience certainly brought into focus some of the potential value of decentralized and distributed clinical trial initiatives, which I'm sorry, don't mean to give a plug for you and your colleagues at Curavit and your other peers in the clinical research world. But some of the initiatives you all are helping to spearhead are really important to allow, hopefully allow at least some study-related processes and testing to be done closer to home, maybe eliminate at least some of those visits, and hopefully decrease the burden of study participation on patients and their family members and caregivers.
Oh, thank you. I appreciate that acknowledgement. How long were you in this trial?
So things remained stable on the trial for 3 months, which in the context of cancer that's a good thing if things are not progressing, in other words. Unfortunately, at the 3-month point, both my blood studies and CT scans showed that the tumor was progressing, the metastases were progressing, and I came off the trial.
So the trial was unsuccessful. That must have been rather devastating news. What came next for you?
Yeah, so there was a procedure called radiofrequency ablation. Again, apologies for the technical details here. In this procedure, a probe is actually inserted through the skin of the abdomen using ultrasound for guidance into the liver, into the metastases, to use electrical current to try to destroy the metastases. And at that time, encouraging information had been published regarding improvements in survival for individuals with colorectal cancer with liver metastases with this radiofrequency ablation, usually abbreviated to RFA
Mm.
procedure. There was really no data in the literature about RFA for gastric cancer liver metastases, but based on the colorectal cancer experience, one of our interventional radiologists was graciously willing to try this in my situation. And unfortunately, although the procedure was effective in destroying a good number of metastases, new ones surfaced very rapidly afterwards.
Mm. What did you do then?
Yeah, so we had been through a trial. We had been through attempted local therapy. So my oncologist, my wife, and I collectively thought it'd be reasonable to try one of the second-line conventional chemotherapy regimens. For this, I returned to the S1 I used initially. We added a different medication called docetaxel, used for many types of cancer. And this regimen actually kept things stable for about 4 months, at which time, again, increase in the number and size of the metastases was noted on my scans.
Mm.
My oncologist then switched me to a third line of conventional chemo, but I was too nauseated with this, really couldn't tolerate it, so we had to abandon that.
So you've been through so much at this point. It seems like options are decreasing. Am I thinking about that correctly? And if so, what do you do?
Yeah, so again, we were looking with my recent experience with conventional chemo, we were trying to see if there was something localized. Again, my cancer was only confined to my liver, which is, in a sense, good news. So we were looking at options for local treatment of the liver metastases. Again, at this time, some encouraging information had been published regarding prolonged survival for individuals, once again, with colorectal cancer and liver metastases after procedures where part of the liver was actually removed, part of the liver containing the metastases. And that was interesting information that that was associated with prolonged survival. You might ask, well, how can you even remove part of the liver? And one of the key things about the liver is it's one of our few organs in the body that actually has the potential to regenerate. So that allows
Mhm.
even a significant portion of the liver to be resected therapeutically. One of our expert liver surgeons was familiar with this partial resection. The technical term is partial hepatectomy procedure for management of colorectal cancer, liver metastases. And he was, again, very gracious in being willing to try it in my case with our mutual understanding that there were no data, again, regarding the potential success or failure of this approach in gastric cancer, gastric cancer, liver disease. Again, I didn't have a lot of options, Dave, as you mentioned at this point,
Mhm.
so I opted to pursue this major surgery. And again, it was effective in removing parts of the involved liver, but unfortunately, as the liver regenerated, so did the metastases.
Mm. And after what seems like an incredibly involved surgery, if I'm hearing this correctly, what did you do at this point?
Yep. So between the RFA procedure, the partial hepatectomy, which was quite extensive, I was in the hospital for a good week after that. My care team and I felt like we had exhausted local treatment options. We decided to head back to clinicaltrials.gov to see if there are any other clinical trials that looked promising in terms of other targeted therapy options. We still thought at that point that the MET pathway that we mentioned earlier was driving the growth and spread of my cancer, although that previous anti-MET trial was effective for only 3 months. It did have some effect, and we thought that perhaps the lack of long-term effectiveness just reflected an insufficient degree of inhibition of that MET pathway by that particular agent. So We hoped at that point that another class of agents targeting that MET pathway might be more effective. One such class of drugs that was under active study at that time were monoclonal antibodies, and we mentioned earlier the rituximab and the trastuzumab. Those were both monoclonal antibodies that were very effective forms of targeted therapy. These are larger molecules. They actually bind to receptors on the surface of the cells, hopefully blocking proteins that are key to the tumor cell in terms of its survival, growth, and proliferation. We were excited in view of that really positive experience with rituximab and trastuzumab to read about a trial of a new monoclonal antibody targeting that MET protein.
Was this a phase 1?
Yep.
What type of trial was this?
This was really new, hot off the press. This was a phase 1 trial, again, mainly geared towards safety and determination of a maximal tolerated dose. This time, the closest trial center was 45 minutes by train south of Newark, New Jersey. Newark itself being a 3-hour flight from Minnesota. So study visits, this time invariably I had to come in the night before, had the study testing and drug administration done and was able to leave the following evening. I did, speak with my wife and kids every night by phone, but I still missed being able to see them while I was away. So I received this experimental drug. It was IV, given once a week in the center for 4 weeks, as is often the case with these studies. Usually at the 4-week point, you have a CT scan and some other blood studies. And unfortunately, both of these showed that the metastases had progressed significantly, and I had to come off this study as well.
Another study and another disappointment.
Yeah, no, this it was certainly disappointing. Not only were we disappointed with the results, but the failure of this second anti-MET agent really called into question for us the whole concept of targeted therapy, or at least therapy targeting this pathway based on those chromosome results and that information about chromosome 7 and the MET protein and gastric cancer. We thought that intellectually we kind of had an understanding of the pathway that was driving the growth and spread of my cancer, and this worldview, so to speak, of my cancer was really called into question by the failure of this second trial.
What did you do next?
Yeah, so at this point, to recap, and I appreciate everyone's patience with this long drawn-out saga here, but to recap, at this point, we were 22 months out after my diagnosis. I had failed extensive surgeries, 3 lines of conventional chemotherapy, 2 targeted agent clinical trials. I didn't mention I had, during the course of this, had a number of major medical issues, including a collapsed lung and a serious spine infection that necessitated emergency neurosurgery and 8 weeks of home IV antibiotics. So not a very positive time. My liver function, unfortunately, the numbers were really starting to deteriorate, and I was actually having more pain at this point. The way things were progressing overall, it looked like that maybe I had one more shot at treatment. We identified on clinicaltrials.gov yet another trial targeting MET. This one was combined with another medication. We thought, well, maybe a combination approach might be more effective. But we wanted to make sure that this would be the most appropriate option to pursue, given that, again, this might well be my very last try at therapy. So it was at this time, serendipitously, and serendipity has played a huge role in my entire care, certainly from the beginning on. But serendipitously, we came across a research article that had just been published by a research scientist specializing in this MET pathway. And this was a preclinical study, Dave. So it was not a study in patients. It was an experimental lab-based study, but his information was very interesting. It was clear that he was an authority on this pathway. I emailed the scientist who very graciously offered to actually study some of my tumor tissue in his laboratory using specialized research techniques that were not available clinically at that point, to determine if this MET pathway was indeed overactive and was driving my cancer. Well, the studies he performed revealed, in fact, that the MET pathway was not overactive, and this meant that further therapies directed towards the MET pathway were not likely to be productive. The scientists, though, very kindly also looked at other proteins that were known to drive cancer progression. And he noted in his testing extensive activation of the HER2 pathway throughout my tumor. And we mentioned this pathway a little bit earlier, but the degree of activation was so extensive on his testing that he concluded basically that based on his testing, it was not the MET pathway, but rather the HER2 pathway that was driving a tumor progression in my situation.
So this sounds hopeful. How did you respond to this news?
Yeah, well, we were, I would say, cautiously optimistic, Dave, at this point. As we mentioned earlier, the HER2 pathway was well known to medicine and oncology. The pathway itself had been identified literally over 20 years earlier. Trastuzumab, which is, again, that monoclonal antibody targeting the HER2 protein, had been approved by the FDA as effective in the treatment of HER2-positive breast cancer, as we mentioned. And parenthetically, for folks who are interested in the drug development process, it took a full 15 years after the elucidation of that HER2 pathway for the targeted agent trastuzumab to be FDA approved. So it's quite a lengthy, convoluted process. And for anyone interested in reading more about that process, I highly recommend the book by Robert Bazell. But anyway, getting back to my situation, at that point, we looked at the literature. We found a single case report, recent report, of an individual with HER2-positive gastric cancer who had been treated with trastuzumab with some improvement. And basically, based on that report and on the results of my research lab studies showing the HER2 activation, my oncologist wrote a single patient expanded access clinical trial protocol to allow me to be treated with trastuzumab. The protocol was approved, had to be approved by our institutional IRB as well as the FDA, and I received my first IV infusion of trastuzumab. 3 days later, I woke up with a massively swollen right arm. I went to the emergency room. Studies showed that I actually had a major blood clot involving my arm and my neck. I was placed on anticoagulation. We decided, again, in view of the lack of other options, to continue on with treatment. I continued to receive the IV trastuzumab weekly for the next month. As we mentioned earlier, in many types of clinical trials, imaging studies are often performed at a month after the initiation of the regimen. And accordingly, at the 1 month point, I found myself back on the CT scanner table. I still remember to this day kind of wondering what this scan is going to show regarding the effectiveness or lack thereof of what was likely, again, my last trial of treatment.
And what did the scan show?
Well, fortunately, the scan showed improvement. Based on those results, I continued the trastuzumab combined with IV-5-FU and another medication called lapatinib. That's an oral agent which inhibits the HER2 pathway, but at a different point compared to the trastuzumab. Lapatinib had been found very helpful in conjunction with trastuzumab for the management of HER2-positive breast cancer. So based on that information, we proceeded with that combination treatment. Overall, I had a dramatic response to therapy, and a year later, had no evidence of cancer on my scans or blood studies, and I've been fortunate, knock wood, to continue to do well since.
I'm so happy to hear that. It's an unbelievable journey.
Thank you.
What do you think worked?
I've been incredibly fortunate on so many levels, and that's a great question, Dave, in terms of what worked. I think identification of that overactive HER2 pathway was key in terms of steering us away from the MET therapy and towards the trastuzumab. I was, again, extremely fortunate that there was an FDA-approved drug available, trastuzumab, to inhibit that HER2 pathway, and that it was accessible to me. It was available to me on this single patient access basis. And again, tremendous gratitude to my oncologist, to the IRB team, to the FDA, as well as the research scientists for their roles in this regard. I have to mention that, as is often the case for folks facing advanced cancer, I was also using several supplements from Eastern and Ayurvedic South Asian medicine that are used for cancer treatment. The supplements I used had actually had some evaluation in standard, quote, Western biology labs, and have been found to have at least a basis for synergistic effect. So I wasn't just taking the IV 5-FU, the trastuzumab, milpatinib. I was doing a couple of other things at the time. I used to think, getting back to the clinical trials, the first two trials I was involved with, I used to think that those trials targeting that MET pathway were not effective, that they weren't successful. But interestingly, about 5 years or so after, maybe 6 years after going through those trials, research was then published revealing that there is a phenomenon called crosstalk, or communication between the HER2 and the MET pathways. And it's felt, in fact, that one potential mechanism of resistance that many patients unfortunately develop to trastuzumab is activation of this interlinked or cross-talking MET pathway. So it's possible in my situation that the blockade of that MET pathway, although it was not sufficient alone to address the growth of my tumor, it may have set the stage for more effective treatment with trastuzumab, resulting in what I, again, am very grateful to say has been a more profound and extended response to trastuzumab in my situation.
That's an incredible story. You've now participated in multiple trials, multiple phases, many failures, one incredible success. As you reflect on your experience as a participant, what were the positive experiences and what might be things you'd like to see improve from a participant experience going forward?
Yeah, great question, Dave. So first and foremost, I have to say my experience initially as a patient participating in clinical trials, and more recently now as a patient advocate supporting patients who are considering trials themselves or are actively participating in trials. Really, this whole process has only served to deepen my pre-existent appreciation as a physician for our clinical trials system. Trials, as we know, and I know this is not information I need to share with your very well-informed audience, but they play a major role in establishing the safety and efficacy of new therapies. They serve certainly as a basis for regulatory approval and distribution of drugs or other treatments. Following approval, the data generated during the trials provides important guidance to individuals undergoing treatment, to their caregivers, healthcare providers, in terms of what to expect with the use of therapy, adverse effects to monitor for, etc. Really importantly, very importantly, for individuals with cancer that has progressed recurred or spread after standard therapy, as in my situation, for patients and for their caregivers, clinical trials serve as a very important source of hope, which is really, I think, essential. So overall, Dave, I am a huge fan of clinical trials. Having said that, I think there are definitely some opportunities for improvement in our current system, many of which I think you and your colleagues at Curivate and others in the clinical research world are pursuing. But from the patient and now the advocate perspective, these include, I'd say, number one, decentralized and distributed trials. Again, I was really fortunate to have the ability to travel to distant sites for my trials. This was associated with significant expense and time away from loved ones. Travel-related barriers may interfere, though, with the ability of patients to access trials that may be helpful for them, and also, in some cases, may delay patient accrual to these trials, may delay trial completion, which is very important to our biopharma partners. This can ultimately impact the overall availability of these important new treatments. Frankly, many, if not most of the tests that I had performed in the distant trial centers could have been done closer to home. I absolutely respect the importance of trial data integrity, test result integrity, but maybe centralized training and accreditation programs could be implemented to assure that each participating center is able to provide reliable data. Things have changed a lot again over the last 17, 18 years. Now with our current levels of health record and imaging system interoperability, where data can be shared instantaneously at a great distance with high fidelity, the results of tests, such tests done closer to home, could be shared quickly with the PI at the distant institution. So that's number one. Sorry, I'll be going on a little bit here, Dave. I apologize. Number two, I would say is standardized and streamlined IRB approval processes. These vary quite a bit from institution to institution. I'm currently, as an advocate, supporting an individual who wants to participate in a particular trial. The trial is open and active at a couple of centers, each of which is 1,300 miles away from his home. The individual's local institution is also listed as a participating institution, but activation of the trial at his institution has been delayed for now a good number of months due to internal institutional and approval processes. And again, I completely understand the desire and the importance of medical centers in terms of retaining their autonomy and all, but it would be wonderful from the patient's perspective if some sort of super multi-institutional IRB comprising representatives from multiple institutions could review a protocol and then allow its activation at the participating centers without further review by the local IRBs. Item 3, terminology, which again I know is familiar to you and your audience, adaptive and pragmatic trials, I think can really be very helpful for individuals facing cancer and other serious conditions. Modifications might include the ability of study investigators to adjust group sizes or even discontinue study arms during the course of the study based possibly on interim data analysis or even surrogate endpoint indicators. I know this is not the forum to discuss all of these technical options. In a nutshell, no patient with a serious condition wants to be assigned to a study group where interim data for that group shows that really suggests futility or potentially even harm. Similarly, pragmatic trial designs where trials are performed in real-world settings may, again, be welcomed by participants, particularly if they're combined with decentralization initiatives. The next item, this is 4 from my bucket list here, would be study group design changes. Again, individuals facing serious diagnoses with a significant likelihood of poor outcomes with standard of care are often understandably reluctant to participate in a randomized controlled phase 3 study where they may in fact be assigned to standard of care treatment. Again, I was very grateful in my 2 trials to know that I was at least getting the study drug. This reluctance, again, I think has translated to difficulty in certain studies meeting their accrual targets, resulting in increased expense to the sponsors, delayed approval, and unfortunately, in some cases, closure of the study before results were even generated. And again, I fully understand there's no simple solution to this problem, but potential patient-centric improvements in terms of study design might include reduction in the size of standard of care groups relative to the interventional groups, maybe use of historical controls that are ideally drawn from the same patient population that is being studied. And certainly, I think early crossover designs where individuals in the control group are offered opportunities at early points in the study to join the treatment group. Again, this may be facilitated by interim data analysis looking at surrogate endpoints. And the fifth thing, and this will be the last, thank you for letting me have this mini soapbox here, Dave.
Thank you.
Is the importance of real-world evidence capture. And as we mentioned earlier, important efficacy and certainly safety data beyond that, which is available from phase 1 through 3 trials, may develop as drug use or intervention use is broadened among the population, larger number of patients, more diverse populations. Again, patients receiving treatments in non-trial settings. This, I think, is important for folks who may be continuing to receive their investigational agents after the conclusion of trials, which, again, I heartily applaud trial sponsors for, in many cases, sponsoring or at least permitting. But patients using drugs on a longer or other interventions on a longer term basis, I think would be reassured to learn that there is a robust data capture system in place to capture and share as necessary data that are relevant to them, either positive or more potentially negative. Another thing which I think is a real potential, really I find particularly exciting, is that real-world evidence generation and capture might also facilitate capture of data from so-called N of 1 trials, such as mine, frankly, which might inform treatment of future patients and even serve as a basis for generation of hypotheses directing future research. So thanks for the soapbox again, Dave. Appreciate the opportunity to share those thoughts.
Well, thank you, John. This interview has exceeded my high expectations going in, and I appreciate your candor, your openness, thoughtfulness, and then particularly appreciate your ideas for future improvement. That's what I think we're all focused on, is sharing the stories of participants like you and focusing on how trials can be made better for future participants. I want to shift gears a little bit. I heard from a colleague of yours that you have, along with your wife, written a book about this. Can you say more?
Yes, thank you. And no, I appreciate your mentioning this. Basically, when I was diagnosed, and especially I would say at the time my metastatic disease was diagnosed and was progressing relentlessly, my wife and I kind of wished we looked and really didn't find much in terms of a step-by-step reference to help guide us. In addition, over the years now, we've shared advice with a soberingly large number of family members, friends, colleagues who have faced their own cancer challenges. And several folks asked, in fact, if we would consider putting aspects of our experience down on on paper are lessons learned, so to speak. And the COVID pandemic came along. Not a lot of socialization at that time, so a little time to write as well. So
the book, I believe, is called Facing Cancer with Hope: Suggestions for Patients, Caregivers, and Patient Advocates, based on a surgeon's own cancer journey. Can you give us a couple of take-home points from the book, and then we'll encourage listeners to find your book?
Oh, well, thank you, Dean. So first and foremost, we have to acknowledge that every individual is different, and this book is not and was not intended to substitute for the medical guidance that every individual should get from their own care team. But here are a few general suggestions based on our experience. First, advocate for yourself or for your family member or friend that you're supporting. That's very important. I think most of us are equipped now with the data resources available online to become a mini expert, as it were, on your type of cancer or your loved one's type of cancer, the molecular biology of the tumor. Many of us, and I'll include myself, didn't get deep exposure to biology or oncology during our formal education, but with, again, the resources such as PubMed, clinicaltrials.gov, Google itself can be very helpful. There are now also available clinical decision support tools that are available to the public as well as healthcare providers such as UpToDate to learn. We can use these resources to learn about our malignancies and related conventional and experimental treatment options. I've been personally very impressed recently by an AI-enabled platform called openevidence.com. I have no affiliation. I'm just a user with them, but it provides organized and very well-referenced answers with hyperlinked reference numbers, so you could click on the reference, look at the original source, to specific medical questions that are based on information in the medical literature. Related to this whole process of doing your research, I think it's important, particularly when you're at a stage where there may not be a clearly defined best pathway forward, to engage in a process called shared decision-making with your oncologist, in which your particular preferences, needs, and values, as well as your medical status, are taken into consideration in terms of making treatment decisions. One small thing that I found helpful was to try to think 2 steps ahead. Grandmasters in chess, I think they can think 12 moves ahead, and Go players 25 moves ahead. I'd settle for 2 steps ahead, meaning have a backup plan in case the current treatment or trial you're on fails. I think this can help allay some of the uncertainty, the emotional disappointment, if you know what you're going to, move on to next. Certainly for all of us, I think at all stages in our lives, we need to be aware of setting or resetting our priorities. For me, concentrating on what I could do and can do to best support my wife and kids really proven to be a very valuable strategy as we navigated my cancer journey. This is trite, but maintain nutritional and safe regular exercise. There is actually a really solid and expanding body of evidence showing that folks, even with metastatic disease, who exercise may do better in terms of survival. So these are things that are still under our control. Do what we can. The exercise should be obviously safe and tailored to your particular situation. I was extraordinarily fortunate to have so much support on many levels, Dave, from friends, families, coworkers, members of one's faith community, can all provide general support and sometimes actually relevant specific advice. Disease-specific patient advocacy groups are another tremendous resource. And these weren't really around for gastric cancer when I was going through the acute phase, but they can provide individuals with access, really very direct access in some cases, to leading clinicians and research scientists. So those organizations are really, really important to keep in mind. finally, to paraphrase Winston Churchill, never give up. Our understanding now of cancer biology, the development of new treatment options, importantly, the innovations that, again, you and your colleagues are pursuing in terms of improving clinical trial evaluations of these options, which will hopefully get these options to us as patients more quickly. All of these things are advancing at a pace that's unprecedented in my 40-year career in medicine. Giving the increasing number of new cancer treatment options, and indeed actually now entire new categories of therapies, including new types of targeted treatments and the whole new category of immune-based treatments, we may actually face, in some cases, a good problem of having a large number of trials to choose from. Again, a good thing. I'm optimistic in this regard that AI-supported platforms such as Trial GPT, developed by the National Cancer Institute, may help us as patients and caregivers and our care teams select the clinical trials that are most relevant for us, and at the same time, helping our trial sponsor partners meet their accrual goals effectively and efficiently. With the availability of new treatments, and I don't know if you saw this article, Dave, in the paper, Wall Street Journal recently, there are actually an increasing number of individuals with advanced or metastatic diagnoses who are living for longer and longer periods, moving from therapy to therapy as time goes along, and that's really giving rise now to the concept of management of advanced cancer as a chronic disease.
Mm-hmm.
So keep going. Don't give up hope.
Well, John, you never gave up hope. You never gave up. And I think not only have you and your family clearly benefited, but I think everyone who listens to this, who's a participant, benefits. I think your book continues to provide those benefits from your journey and your efforts. So thank you for sharing your story with us today. And thank you for never giving up.
Oh, and thank you, Dave, for the chance to visit today as well.
The participant story is brought to you by Curevit Clinical Research. Curevit leverages technology, local healthcare infrastructure, and decentralized clinical trial elements to design and execute clinical trials where participants come first. Learn more about Curevit and their innovative virtual clinical site at curevit.io.